Welcome everybody for this late Friday evening talk. For Samuel, it's more lunchtime talk that he has right now. We invited Samuel here because he has done some very exciting scientific work on the bees are not feeding on hemolymph. And as I told Samuel earlier today, I'm pretty uh, um, I'm not happy with that because I have been teaching you beekeepers for more than 25 years that the bees do feed on the hemolymph, or that the Varroa do feed on the hemolymphs on the honeybees, and that's really why you're here. And it's Samuel Ramsey working at the University of Maryland and at a Van Engeldorf bee lab, as far as I understood you call it. And Dennis Van Engeldorf, he's a good friend of Danish beekeeping, and he actually turns up in Denmark once in a while because he has some connections and some good friends here. Samuel, you just finished your PhD recently, I'm sure, on this subject here, so
Thank you very
congratulations
much.
on that. Yeah, and you have been you're really looking on the bee parasites
Mhm.
with regard especially to the varroa mite, which
Yes.
is the major problem, and then on Nosema. So please go for it and correct me
No, you presented
everything I
that
didn't
very well.
present in the
I
right
have
way.
not been doing as much
Welcome.
work on Nosema now. I've shifted a lot of my focus. I'm focusing on Varroa and Tropicolaps mites because I do believe that Tropicolaps will be the next big problem for Western beekeepers. So, uh, yes. So, do you want me to get started now or? Great.
Yeah,
Okay.
yeah.
Well, then here we go. So uh, as he's already explained, I mean, he kind of gave away the end of the movie, but so does my title. Varroa is not feeding on the hemolymph of the bees, and this is something that in the Van Engelsdorp lab, I've been working on this project for a few years now. And with each year, I learned a bit more about how the parasite impacts the bee and what tissue they are feeding on. And I think it will be I think this information will be very helpful for beekeepers to understand how to use this in the future. So, um, wait, there we are. Okay, so Varroa needs no introduction, so I won't spend very much time on this slide. Varroa is the biggest issue in beekeeping because a lot of other issues connect to Varroa. Their feeding causes the bees to be more susceptible to pesticides. It reduces their immune function. So even though there are a lot of other problems like parasites or like viruses and bacteria and pesticides, Varroa seems to be the linchpin issue. They were first found they started spreading internationally about 60 to 70 years ago. They started moving around the world. They have achieved a nearly cosmopolitan distribution. They are almost everywhere in the world except Australia. And so they're causing a lot of economic injury. But the reason why this map is very important is because the very first areas where Varroa started to spread were China and Russia. And as a result of this, the fundamental information about Varroa, uh, all of the basic things that have been written about their biology are written in Chinese and they are written in Russian. And so that's one of the reasons why uh, we actually don't know what the parasite is eating when it feeds. We were told that they feed on hemolymph in a paper that was written in 1972 by a Russian researcher. And the only part of the paper that was translated into English and other languages was the abstract, which is just a one paragraph summary of years and years and years of research of everything that that paper encompassed. And so, because of that, the abstract summarized things in a way that made people think that the paper had proven that Varroa feed on the blood of the bees, the hemolymph. But this paper unfortunately did not show that. All that it showed was the amount of food that the mites consumed. It showed that they did consume a substantial volume of food, and the researcher assumed that that food was hemolymph. So since then, we have been repeating this because it was eventually cited in a paper, in several papers in the US and in Europe, and everyone started citing the researchers who cited those papers, and not a lot of people have gone back and read the original resource. And so it's caused a lot of confusion. And as a result of that, I decided to take on this project because the idea that these mites were able to reproduce so quickly with just hemolymph did not make sense to me. Hemolymph is mostly water, and the mites' eggs are very large, and they make a very large egg every 30 hours. It is almost 40% their body's volume, and so they need a lot of nutrition to do this. So when I started this project, I really wanted to make sure that it was something that would pan out. I needed to be sure that I could finish this project and still get my degree with it. And so I looked for some expectations that I would have. If the mites are actually feeding on hemolymph, I expected that these things would be clearly the case. If the mites are not feeding on the hemolymph of the bees, then these expectations should be incorrect. And so I started with these. The first one was I did not expect the mites' digestive system, or sorry, I did. I expected that the mites' digestive system and their excrement would be very similar to the digestive system and the excrement of other insects and mites that feed on the blood of their host. And this was not the case. Varroa's excrement, it's this white, let me see if I can move the pointer to it. Do you see the white stuff on the inside of the cells? So this is the excrement of the mites, and it is made of guanine, very concentrated guanine. And guanine is a constituent of organ meats. You'll find it a lot when you break down the meats through catabolism. Guanine is the waste product that is typically produced. But that's not what you would expect to find a lot of in the blood of the bees. It's heavily nitrogenous, and you don't find this in the bees' blood. So it was very interesting to see that, and that was from research that was conducted by Dr. Alan Cohen, who has been skeptical for some time now that they could get this much guanine from the blood of the bees. Another thing that is very strange is the digestive system of the mites. Their digestive system is not shaped correctly for them to feed on the bee's blood. This is the digestive system of bedbug or an aphid, insects that feed on something that is mostly water with very little nutritional content in it. In order for those creatures to feed on these things and get enough nutrition without swelling up their entire body and potentially even popping, they need a system that will exchange all of the water out of what they're eating with their rectum. And so here we have the digestive system. This is the esophagus of the bedbug, and this is where it touches the rectum. So if you see this section here, it touches the rectum right where it comes into the body, and it exchanges all the water out of the bedbug's body, or out of the bedbug's meal, so that it can concentrate all of the nutrients from what it's consuming. And we would expect that if Varroa is feeding on the blood of bees, which is mostly water, it's even a higher water content than what bedbugs feed on, human blood, we would expect then that the digestive system would have a similar shape to this in what we see with all of the rest of these organisms. Now, Flemming, could you pull up the video? Thank you very much.
Correct.
This is what the digestive system in Varroa looks like, and please excuse the leg is kind of getting in the way, but if you'll look right here, I wonder how well you can see this. Inside of the mite's body, you can see this peristaltic motion. There is just one tube that runs to the end of the mite's body. You can stop the video now. Thank you, Flemming.
But it is having to feed on
that section of the digestive system, and you know what, I can point to it right here. So the mite's digestive system is this white tube that runs from the front of its body all the way to the back, and this larger section right here, that is the rectum of the mite. This is where you would expect the fluid to be excreted. If the mites are feeding on something that's mostly fluid, they have to get rid of all of that excess water while they're feeding to concentrate the meal. The mice's digestive system never loops around so that the esophagus touches the rectum. And so they don't have the kind of digestive system that you see in creatures that are processing a meal that is mostly water. So that was immediately something that had us thinking that there was more going on here than the hemolymph hypothesis.
Excuse me,
And
Samuel.
so,
Samuel, would you please start your webcam again? We want to see you when you're talking.
yes,
Shut up. Thanks.
thank you. Totally didn't notice. All right. The second expectation that I had was that the mites' lineage, their evolutionary history, would show that they are closely related to other creatures that feed primarily on the blood of their host. Usually in evolutionary processes, it takes a lot of evolutionary time to change your entire digestive system, the way that your mouth parts function. And so creatures that are very closely related often have very similar feeding abilities and behaviors. And so we would expect that these mites would be closely related to other mites that are feeding on the blood of their host, or at least some sort of very dilute fluid to consume. But we do not actually see that. What we see is that the mites are very... so this is a phylogenetic tree. I know it is very large. At the, let's see, this section of the tree, we have the mites that feed on... Is the arrow working? It is not. There we go. On this section of the tree, we have the mites that feed on the blood of their host. We have ticks, which are also mites, the argacids and several other ticks. And the rest of these creatures do not feed on the blood of their host. And all the way down here at the bottom we have Varroa. It's very, very, very distantly related to ticks and other creatures that feed on blood. But the creatures that it is very closely related to, and this is that same phylogenetic tree, I've just zoomed into the bottom. It is very closely related to the Gammacyphus, Macrocheles, Lasiaceus, and Cosmolelaps mites. And all of these mites, please remember those names, they'll be on the test at the end. All of these mites are creatures that feed on their host through a process called extraoral digestion. Instead of feeding on just the blood, they stick their mouth parts into their host, they release digestive enzymes, and it breaks down the host's insides, or a section of the host's insides, and then they suck that up. And what we found is that Varroa's digestive system is very similar to the Cosmolelaps and Macrocheles mites, which means it is very likely that Varroa feeds in a similar way, using extraoral digestion to break down some part of the bee and then feed on that instead of the blood. So those first two expectations that we would have, if the mites are feeding on the blood of the bees, those are not what reality shows us. These are not accurate things that we see. So it is very possible that the mites are not feeding on the bee's blood. Well, my third expectation, I could not look at other literature to find the answer to this. I had to do an experiment, or at least an observational study myself. And so my study was designed to find where the mites feed. Because if the mites can feed anywhere on their host, the way that maybe a mosquito or a tick can with you, it's probably feeding on the bee's blood. Because a mosquito or a tick, they can feed anywhere on you because blood is ubiquitously distributed inside of your body. You have blood everywhere. The bee also has blood everywhere. And while there are some areas that it may be easier to penetrate the bee than others, we would not expect that if they're feeding on blood that they will only be able to feed in one location. But if they do feed in just one location, maybe they are feeding on a tissue that is only in that location. And so the experiment was very simple, or the study was very simple. I went into the brood nest of several colonies that were not treated for Varroa, and I would grab the nurse bees from the brood nest, and some foragers sometimes too when they were there as well, and we looked at where the mites were on those bees. So we would grab them by the wings, turn them over, and record where the mites were on the bees, and we used this as a data sheet. Wherever the mites were, we recorded it on here, and this is the results of that particular study. What it has shown is that even though, and this is a heat map. So with just a bit of explanation, I believe it's very straightforward, but the bluer regions are regions where there are very few mites, and the red regions are regions where there are a lot of mites. And so this section of the mite's body is, of course, the thorax. The thorax is the place where I always see the mites in pictures. Now, whenever you see pictures of the mites, this is the area where you see them. But as you can see, less than 5% of the mites were found in that area. However, 95% of the mites were found on the underside of the bee's abdomen, in this region. And it was very interesting to us to find this out, that the mites very strongly prefer to be on the underside of the bee's abdomen, because we show these images so frequently that I think we have people convinced that this is where the mites are normally found and this is where the mites normally feed. When I give these presentations in the US, I give them to a lot of beekeeping groups, and we also go there with something called the National the National Loss Survey and the Management Survey. And we ask these beekeepers a lot of questions. One of the things we ask is, do you treat for Varroa? And most beekeepers, close to 60% of beekeepers, do not treat for Varroa. And so we ask them, why? Why don't you treat for Varroa? And they tell us we don't see them in our colonies, and so we don't treat for them. So then my next question to them, actually we have it on the survey. We say, how are you monitoring for Varroa? Do you use a sugar roll, an alcohol wash? And they will usually draw a third box and write other and say, I do visual inspection. And so then we have to ask, where do you look on the bees for the mites? And they say, on the bee's back. We watch them walk around on the frames, and if I don't see any mites, I don't treat. And the problem is that if that is where you're looking, you will never see the mites, because they are actually much more likely to be found here. 95% of the time in our work, we have found them on the underside of the bee's abdomen. They tuck themselves between the segments that make up the bee's body. The bee's abdomen is made up of a bunch of plates that overlap, and the mites tuck themselves between those plates. Flemming, could you do the other video as well? Thank you very much. So this is an electron micrograph of a honeybee's plates. And in the middle here, you can see that there's this red section. That is the Varroa mite's body, and it's tucked between the two plates of the honeybee's body. So it's very interesting how the mites have sort of blurred the line between being an external parasite and an internal parasite. Even though they're technically on the outside, they've wedged themselves, and their feeding is internal. All right, thank you.
And sorry, then you have to turn your video on again.
Okay, so we thought that was very important, but because this work is it is overturning things that people have believed for a long time, we received pushback from researchers. They wanted to, and I appreciate this from them, they wanted to be sure that we were doing our due diligence. Since we are attempting to overturn work done by other people, we can't do what they did. And a lot of these people made a lot of assumptions, so we will stop, we will try our best to not make a lot of assumptions ourselves. And so the researchers asked us, how do you know that they're feeding in this area? Maybe they just like to hide here. Maybe there's a good temperature, a good humidity. Maybe the bees are, maybe the mites are wedging themselves in this area because it is more difficult for the bees to groom them away. And so what we needed to show was that this is actually the region where feeding occurs. When we removed the mites from the thorax of the bees, we saw no evidence of feeding. What we saw was questing behavior. The mites, when they are on the thorax, when they are on the bee's back, they raise their front legs and they wave them about, and it is a way of them looking for other bees, because remember, the mites are blind. And so they use their front legs to smell things around them and to touch things. And when they find a new bee, they can attach to it. They do this when the bee that they have been feeding on is so damaged that it cannot sustain them any longer, or when they need to get on a forager and leave the colony because the colony is dying. And so that is why it's it is not a good thing that so many people do not treat for Varroa until they see them walking around on the bee's back, because oftentimes those bees are so unhealthy now that the mites have to leave the colony, and so it may not help at all to treat at that point. The bees may be dead and just not know it yet. If we're going to determine whether the mites are feeding, we had to be able to remove them from the abdomen and look for evidence of feeding there. And the problem is that the membrane that is between those two plates is very, very thin. It is 15 micrometers thin, and that is like if you take 15 bacteria and you stack them up end to end, that's how thick this membrane is. So very, very thin. And anytime you try to move those plates apart from each other to see what is between them, the membrane tears. And so you can't tell if you caused the damage or if the mite caused the damage. And so what we did was Faradic, oh, okay. I'll get back to that in a moment. So what we did was we froze the mite in place, and we used liquid nitrogen to freeze everything in place immediately. And this was really helpful because by freezing them in liquid nitrogen, everything stops. It's down to negative 196 degrees Celsius. And so the mites were frozen onto the bee's body. And so you have, let's see, this is Varroa right here. This is the Varroa's body. And this is the honeybee's plate, called a sternite. And this is the second plate under it, also called a sternite. And what we wanted to do was to remove the mite and see what was under it. And so if you look closely in this area, you can see some very interesting things. Do you guys see what's going on here? Pay special attention to this object and this object, because they will help you figure out what is going on here. It's difficult to see in black and white, so I will add the color back to it. And what you can see here is this is one of the foot pads of the mite. When we dislodged it using the liquid nitrogen, hey, the arrow, come back. There we go. When we dislodged the foot pad, or when we dislodged the mite using liquid nitrogen, and when we broke the plate off, the mite's feet were held in place so well that the mite came off. And so you can see the indentation of the leg all the way across, and then it goes all the way to this object in the middle. And this thing in the middle is a feeding hole. It's shaped exactly like the mouth parts of the mite, and it goes all the way through the membrane to the other side. And so we know that this is actually the region where the mites are feeding. But we also know something else very interesting that came from this. Several researchers told me that Varroa do not feed on adult bees. And they told me this because we call the stage when the mites are on adult bees the Faradic phase. And it was named this because it was named this during a time when we only thought that Varroa fed on the larvae, on the brood of the bees. And so that immediately prompted the question for a lot of people. If they are Faradic, then they do not feed on the adults, because the term Faradic is very specialized. All of these creatures that you see here, these are what are called Faradic mites. They attach to an organism, usually an organism that has wings, and that creature is able to fly them somewhere else. So these are a whole bunch of mites on a beetle, and these are a whole bunch of mites on a dragonfly, and all of them are Faradic. This is a close-up of the mites that were on the beetle. And the question of whether they're Faradic is connected to just one particular thing, really two, but is the creature able to feed on the organism that is moving it around? Foracy is a special parasite term. Only creatures that do not feed on the vehicle that is moving them from one place to another are considered Faradic, because any creature that is a parasite could be considered Faradic if we include feeding. Every time a mosquito lands on you, if you walk while it is on you, it would be considered Faradic. And so if Varroa are truly Faradic, as the term means, then they are like these creatures here. They are attached, but their mouth parts are not actually feeding on the organism. But what we found through our research, we found conclusive proof that they are not Faradic on adult bees. And as a result of that, we have to change the words that we use when we talk about this segment of their life cycle, because this is not Foracy. They are feeding on the adults and causing problems to the adults as well. And so we looked even more into this using transmission electron microscopy. And what we can see here, this is one of the plates of the bee. This is the Varroa mite. This is fat body tissue inside of the mite, and this is the wound that the mite makes in the membrane. And what we found so interesting was that when the mite sticks its mouth parts into the bee and creates this wound, it removes a whole section of tissue that is normally in the body, and it is called fat body tissue. Normally, this tissue that you see over here, this fat body, extends all the way under the membrane. But when the bee has been fed on by one of these mites, the whole section of the tissue is missing. But what we find in place of that is this stuff right here. And it's actually the broken down material of what used to be fat body cells. And they've been broken down through a process called extraoral digestion. The mite is releasing some sort of digestive enzyme, and it then breaks down the cells that are in that area. And so it was very interesting to find that. We then did we took a bunch of bees that had mites feeding on them, and we froze them in liquid nitrogen. And then we did something called a freeze fracture. We took a blade and cut right down the middle so that we could see inside of both organisms. And so this is Varroa Destructor right here. Hey, arrow, help me. There we go. This is Varroa Destructor right here. And this is the inside of the honeybee's body. So that is the top plate of the bee's body. This is the bottom plate that the mite is then standing on. And this is fat body tissue that is inside of the mite, or sorry, inside of the bee. And so it's very interesting looking at these images and seeing the proximity. We know that what the mite has access to when it wedges itself between the plates, it has access to a lot of fat body tissue that it can consume, but not quite as much hemolymph as we've been thinking all of this time. So what is fat body? This is what fat body tissue looks like inside of a honeybee. So all of this, if you open up a bee, the bee's abdomen, and you cut down the side, you open it like a clamshell. After you've removed the digestive system and the connective tissue, all that you will see will be this squishy yellowish white stuff, and this is fat body tissue, and it is wrapped all around the bee's body in the abdomen. So it is on the top side of the abdomen, the bottom side of the abdomen, but on the bottom of the abdomen is the largest layer of it. This section here, all of this. And so this is what it seems that the mites are actually impacting when they feed. But to be sure of this, we really had to do more work. So I've already shown, based on these, the work that's been conducted, that hemolymph is not what they are likely feeding on. But we wanted to be clear as to whether the mites are maybe consuming both hemolymph and fat body, a lot of hemolymph, a lot of fat body, what exactly is going on. And so the next part of the study was to actually take fluorescent chemicals that will actually glow inside of the bee's body and inside of the mite's body. And if we can stain the hemolymph and the fat body with two different colored fluorescent chemicals, based on which one ends up inside of the mite, or what proportion of the two end up inside of the mite, we can tell what the mite is then feeding on. And so we took uranine O, which is yellow, to stain the hemolymph, and Nile red to stain the fat body. And they are inside of these cups. These are our colony cups, and the bees are able to feed on the these glowing chemicals. So the yellow one is uranine. Why does the pointer keep doing that? Okay. There we go. The yellow one is uranine, and this purplish red one is the Nile red, and that one is going to stain the fat body. This one will stain the hemolymph. And I think you can see in this. So this is the abdomen of a honeybee that has been fed on the two different chemicals. And so the hemolymph is yellow, and the fat body is red. And there is so much hemolymph that it can be hard to see the fat body tissue in here, but all of that red section, all of that is fat body that has been stained. And this is what the mites look like after they have fed. So Flemming said that when you give presentations to beekeepers, sometimes it is difficult if you show them a lot of graphs. So I like showing a lot of pictures. I have a lot of graphs to represent this information too, but I think this is a good picture that shows just how brightly The mites glow in one color when they fed on the bees. They glow with the color associated with the fat body, this very, very, very red biostain. Thank you, Flemming. The mite was glowing so brightly that it overexposed the camera. We had to decrease the exposure so that we could see the detail. And when we could see the detail, we could make out the entire digestive system of the mite just in this red fat that's inside of its body. And so that this weird sort of mustache-looking object that goes around the midsection of the mite's body, that is its midgut, and all of it can be made out in the fat body tissue that it has consumed. This is a confocal microscopy image, so it shows even more detail. And you can see all of the corpuscles, the blobs that the fat coalesces into inside of the digestive system of the mite. So you can make out all of that red. This is what the digestive system is shaped like. However, we wanted to be sure that the red wasn't so bright that it was somehow obscuring the hemolymph that maybe the mite was consuming. And so we fed the mites just the hemolymph biostain, not the two of them like we've been doing earlier. We did a second round of these experiments where we fed them just the hemolymph biostain or no biostain at all. So either the one that stained the blood or nothing. And this is what the insides of the mites look like if they are given just water. So you can't make out the digestive system. There's very little that can be seen here. And this is what it looks like if you feed them on hemolymph that has been stained. And once again, you really can't see much of anything. It's because very little hemolymph, if any, is consumed. I've compared it to eating a raw steak. You want the meat, or an undercooked steak. You want the meat, but there's a little bit of blood in it too. And so the mites consume some blood, but it is very, very little by comparison to the large amount of biostain that we see from the fat body. And so I'm going to need to start wrapping this up. I don't want to go on too long, so I'll explain the last experiment, and then I will end my presentation. But I do want to explain something about the fat body. When I first started giving this presentation, I would say fat body, and other people would think that I was saying body fat in a weird way, and so they thought that thank you, Flemming. They thought that the mites were simply feeding on fat tissue, simply feeding on the body fat of the bees. But fat body is different from body fat. Fat body is an entire organ that has 9 different functions inside of honeybees. And so it is related to growth and metamorphosis because the young larvae, they need a lot of fat body tissue to organize their new tissues as they're developing into adults. They need a lot of healthy fat body tissue. So when the larva is transitioning to being a pupa, it is full of all of this white, creamy substance. All of that you see through the larva's skin is fat body. And when the bee's body, the larval bee, when it's being broken down and eventually restructured as a pupa, eventually into an adult, all of those pieces of the broken down insides of its body are absorbed into fat body cells, and then they are slowly released over time so that they can structure the adult bee's body. If you remove fat body tissue during this portion of their life cycle, it will lead to the bees having deformities in their body because they are missing whole sections of proteins and things that they need to make up the rest of their body. Because everything that will eventually be an adult bee is inside of that larva. And so when you remove small parts of it, even little parts, you have removed a lot of what is needed to make the adult bee. So the fat body is very important to growth and metamorphosis, to nutrient storage and metabolism. Of course, it is the place where energy and excess nutrients are stored inside of the bee's body. It allows the bees to fly long distances because they can get that energy from the fat body. And it also is the tissue that detoxifies pesticides. A lot of people have been asking, how come the bees were so much more tolerant of pesticides 20 years ago, 30 years ago, 40 years ago? And now some of the same pesticides that they've been exposed to for decades are now becoming lethal to them in amounts that were not lethal before. When the mites feed on the bees, they damage the tissue that normally protects the bees from small amounts of pesticides. The fat body is able to release specialized cells that will actually pull in the chemicals that can harm the bee and coat it in an oil that will protect the rest of the bee's body from being attacked by this pesticide. When you damage the fat body, then it's not able to do this as efficiently as it would be otherwise. So all of these things, all of these different functions that I've been talking about, every single one of them, all of the 9 functions of the fat body are all impacted by Varroa's feeding. And a lot of people have wondered, why are the bees so sick? How come there are so many different problems that seem totally unrelated going on with the bees? I will not say that all of them are a result of this, but a lot of things that have seemed unrelated to different things, a lot of them are related to the fat body tissue. Because the Varroa release these digestive enzymes, even though they don't eat a huge amount of fat body, the digestive enzymes continue to break down tissue even after the mites have stopped feeding. And so it can damage much more fat body than the mites actually eat. Oh, and I should also say immune function is part of the fat body. It releases the antimicrobial peptides that attack viruses, bacteria, and fungi that get into the bees. So the nosema, the viruses, all of those things would be affected by whether the fat body is healthy or not. And also the vitellogenins inside of the bee's body. These are what allow the bees to extend their lifespan through the winter, so they can live much longer than they would be able to normally, because the vitellogenins decrease oxidative stress. Those are made in the fat body, and they are stored in the fat body. So by damaging the fat body, you also damage the bee's ability to extend its lifespan through the winter. So this is the last experiment that I conducted, and the question that I wanted to ask for this last experiment was, do the mites need the fat body tissue that they are consuming? Is that what it is that they feed on the bees to consume? Because I knew that when we submitted this paper for journals and for reviewers, they would have they would want to make sure that we have dotted all of our I's and crossed all of our T's. And something that we thought reviewers might want to ask is, how do we know that the small amount of hemolymph that they consume isn't what they actually wanted? Maybe because they feed in this area, they get a lot of fat body into their mouth, but it is not what they want to eat. And it's actually the hemolymph that they want to eat. And so they get just a little bit of that every time they feed. And so we conducted an experiment to actually feed the mites the tissue that we want the mites to eat, and then determine the survivorship of the mites and the fecundity of the mites, how many eggs they are able to produce when feeding on a specific diet. And this was this was a daunting, intimidating task to undertake. My advisor was very concerned when I told him I wanted to do this, because a lot of researchers have tried to feed Varroa away from the host, away from a host bee, and it never works. The mites will always die very quickly. And so it was a long shot, a big concern that this would not work. But I decided to try it anyway. And I tried it many different ways, many times. But the goal was to feed them 100% fat body, 100% hemolymph, half and half hemolymph to fat body, or 1/4 to 3/4 of one tissue to the other. And so first I had to build a small little home for the mites to live in. So I created these mite hotels. They're little compressed wax cells that I have hollowed out to be the same shape as drone cells. And inside of these cells, I placed a decoy bee. So if you look here, one of these things is not like the other. One of these things is not a bee. This is a worker pupa, a worker prepupa, and this is my decoy, sorry, this right here is my decoy pupa. And it fooled the mite into believing that it was a bee. It is the same size as the pupa and larvae, as you can see. I've also wrapped it in parafilm. Parafilm is a wax product, but when it's stretched very thin, it feels like the membrane that is on the outside of the bees. It feels like their skin. So it gave them the right touch, because the mites, they navigate based on touch and smell. But the thing that I had to really do to make sure that the mites fed was to actually rub the parafilm against actual larvae and pupae, so that it would have the same smell as the larvae and pupae. And then the mites started accepting it as a potential host. And so then we cut out a section on the bottom of the pill, stretched a very thin membrane over that pill so that the mites could then feed through that membrane. And then using a syringe, this syringe here, we then withdrew hemolymph from bees and injected it into the pills. We also withdrew fat body, so this is a dissected abdomen. We pulled fat body out of the abdomen, and using this, this is a dissected abdomen. Using this tissue grinder, we broke down the fat body into a fluid and injected that fluid into the pills that had fat body in them. And so this is what it looks like. This is our little mite hotel, and you can see the mites inside of here. We used cling wrap as a lid so that we could see directly through it. So you've got the mites, this is the decoy inside, and the results, sorry, but we have a graph. The results are that when the mites fed on fat body tissue, any amount of fat body tissue, they produced eggs. However, when they fed on hemolymph tissue, the blood of the bees, they produced very, 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 very few eggs. Actually, no different from the group that was starved. There was no significant difference between these two. However, there is a very large difference between the mites that were fed fat body, 100% fat body. Those bees were the ones who produced the most eggs, and then 75% and 50%. And what you'll see here is this weird discrepancy. How come they produced more eggs on 50% than they did on 75%? And part of that is an issue well, it's a discovery. If you feed them 75% hemolymph to fat body, the hemolymph and fat body together in that ratio produces a lot of fungus. It is a very good growth medium, and the fungus actually kills the mites. And so there's a group of researchers now working on figuring out if they can use this fungus as a biological control against the mites. But because this fungus killed so many of the mites in this section, in the 75% treatment, we have a big dip here. But pay attention to the 100%, the 50%, and even the 25%, and you see that eggs were produced in substantial amounts in these treatments. And this is the egg of mite. It's very large by comparison to the female, and you can even see the legs through the egg. And this is a survivorship curve. And I will just point out a couple of things on this curve, and then I'm done for my presentation. This shows how the bees survived in the study. And the ones that were starved, it's not surprising that within 24 hours, 80% of them were dead. But what was very interesting was that the same amount in the hemolymph was dead. By 48 hours, everything that was starved or given hemolymph was totally dead. There was no difference whatsoever between the two treatments within 48 hours. However, the only ones who were able to survive the entire 7-day duration of the study were the ones who were fed fat body. And even some that were only fed a very small amount of fat body were able to survive the full 7-day duration of the study. Now, we did still have a lot of mortality across the board because it is difficult to keep these creatures alive inside of an artificial environment, but this is the longest amount of time that the mites have been kept alive off host, and they always survived better when fed 100% fat body without any hemolymph present at all. So these findings indicate very, very strongly that the mites are not using the hemolymph as their primary resource for their food. And so this gives us some important things that we should consider. It's very important that if we are treating for the mites, that we make sure to treat well in advance of the winter, because if the mites are able to damage the winter bees that are being produced in late summer, those bees are going into the winter with damaged fat bodies. And because their vitellogenins have been removed, some of them have been removed, the mites will not be able to survive as long during the winter. And so we encourage beekeepers frequently treat when it's very close to winter so that the mites do not go into the winter with the bees to then feed on them. But what we need to also be thinking about is treating in late summer to make sure that those levels of mites do not get high enough to damage the bees who will be going into the winter. In addition to that, supplementing with protein is not actually that great an idea if you're not going to treat for the mites. If you have high mite levels, they've removed the tissue that stores excess protein inside of the bee's body. And so when they feed on these protein patties, they just excrete all of the excess protein, or a lot of the excess protein, because the tissue that maintains the protein, the excess protein inside of their body, has been removed or damaged. And also there are a couple of groups now working on trying to use this, the information presented by the study, to create a systemic pesticide that can attach to the fat body of the bee and kill the mites when they feed. So thank you very much for listening. Please forgive me for going very far over the 25 minutes. I think I went 40 minutes, so I apologize. Oh, and one more thing, Tropale Labs. Now I am doing some work, or I will be doing work soon in Thailand to see if Tropale Labs is also feeding on fat body tissue, because when I was in Thailand several months ago for part of my thesis, the researchers told me, we believe that they feed on Hemolymph because Varroa feeds on Hemolymph. And I told them Varroa does not feed on Hemolymph. And they said, well, we just assumed it about the mites. We have never tested it. And so because this Tropale mite is killing so many colonies in Southeast Asia, it's now totally outcompeted Varroa, and it's spreading around the world. I have concerns that they will end up in the US, and we won't know how they're feeding or a lot of other aspects of their biology. So that is the future direction for some of my work, is to understand more about this parasite as well. So thank you very much for your time. I'd also like to thank the Van Ingelsdorp Lab and the United States Department of Agriculture. The USDA helped with all of those amazing images you saw earlier. They let me use their electron microscopes, and Project Apis M for funding all of this. So thank you very much.
Wow, thank you. This was amazing, I have to say. Great, and do not worry. That's the great thing with online webinars. I mean, we can keep going all night long if we want to, and people, they can leave and we won't notice it. So if there's really something very exciting to talk about, then I think it's important to just keep going. Lots of questions during your talk. So I don't know how to begin. There was just one here. Yeah. This is Børge. That's a Danish name. Really kind of taking this issue up, that this is not the first time within scientific history that something is overquoted. I mean, it's kind of people are just quoting an article and then they just keep, because it's accepted and because they kind of need to have this reference list. So that's how it is. Let me give you some of the questions. I will start from the end.
Mm-hmm.
And since somebody were complaining in the last talk that I was making too much noise with the paper, my paper is still in my... So this is Christian asking you really about, does this have any kind of influence on treatment in the future? I mean, the mite is always on the belly of the bee, you could say, underneath the bee. Would that have any influence on, let's say, a powder sugar treatment, their oxalic acid trickling, because we really kind of hit them on their back, and even ultrasound. And Christian, I have to say sorry, I forgot to ask about ultrasound for the former speaker. So if you have any opinion about ultrasound, please say something about that. But what does this have as a consequence on treatment? I mean,
remember the previous. Oh, you don't have to remember. I can show you right now. So if you look on screen, remember that the mite's feet, they attach very well to the membrane that is in that area, and so the mites are able to hold on very well. Think about those foot pads like suction cups. I don't know if you use these in Denmark, but in a lot of people in America will actually stick this suction cup to the window of their car and then put their phone on it. And what they find is that if you get any dust on the underside of the suction cup, it slides right off the window. That is the same idea with the dust, the powdered sugar that we use on the bees. The powdered sugar gets in between the foot pad of the mite and the membrane of the bee, and it makes it very difficult for the mites to maintain purchase when attached to the bees. And so they slide off when you shake up the container. And even though they will try to climb back on, the bees are very agitated because of all the powder on them. And so they quickly groom themselves and they knock the mites off. And so then you can use that powdered sugar treatment or that powdered sugar monitoring as a way to tell how many mites you have in your colony by taking 300 bees and shaking them with powdered sugar. It does not work very well as a treatment to control the mites because you would have to get a lot of sugar into a colony all over everything, and it's just very difficult to get an even coating of sugar all over everything. There are people who really want to, though, because it's organic, it's very nice, it's just sugar. But the chemicals, several of these chemicals work, they do still function very effectively, like amitraz. Within the US, amitraz is the only, or at least it is, actually, I do believe it is the only of the synthetic acaricides for which there is very, very, very little resistance from the mites so far. There will be resistance in the future because there is no pesticide pretty much that an organism will not develop some level of resistance to. But these sorts of pesticides absorb through soft tissues, and so the foot pads of the mites and things, as the bees walk over different surfaces, they will get this on their bodies, and as the mites move around, they will absorb it into their foot pads. It will eventually paralyze them by messing with the flow of ions between their nerves, and then the mites will not be able to move anymore, and they will eventually die. And so they are still very effective. There's also the organic acids, formic acid, oxalic acid. There are a lot of options. They are not, I would not say they are directly affected by where the mite is on the bee, because especially with vapors and things, the way that they are released into the colony, as they settle on different objects, they're going to impact pretty much every section of the bee's body. Nope, can't hear you. Flemming, are you talking? I can't hear you.
Because the audience told me to shut up my microphone when you're talking. So, this is another thing. You know, we try to monitor the number of mites in bee colonies. We have the natural mite fall. You could say we have the washing soap method. We have the powder sugar methods, and so on and so on. But now new technology is kind of coming into the picture, and one of the dreams are really, I have a cell phone, I take a picture of the bees, and then I have a kind of a picture recognition something, and then it will tell me how many mites do we have. And there's actually a big Swedish project, and they got quite a lot of money, where they take pictures of the frames, of the back, of the bees. And I can see on your face, please comment on that. This is either you are wrong or they are wrong. I have to say, and it's a pity that so much money is invested into a kind of a project. If 90% of the mites really are underneath all the time, or are they? Yeah. Please comment on this, and I have another question then afterwards.
I shall. Unfortunately, because so many of the pictures that have been shown of Varroa Destructor, the ones that look the most natural, and so the ones that become the most, the ones that you see the most frequently are pictures like this. They look very natural. It's a bee standing up, there's a mite on its back. And this is what people think that they should look for. And so a lot of companies that are trying to find ways of visually inspecting for the mites, they think that it is already clearly proven and shown and known that this is the area where you will find them. There have been other studies in the past that have shown that the mites will actually wedge themselves between the plates of the bees. The very first time Varroa was described by, I believe, a Dutch researcher, Ademons, he said that they have the perfect shape in their body to stick themselves between the plates of the bees. So we've known for a long time that they stick themselves between the plates. What we didn't know until this study is that that is where they are the vast majority of their time. When they emerge and they hang out on the thorax, that is a time where they are showing only questing behavior. And questing behavior is the behavior that you show when you need to get to a new host. And so when they are trying to transfer either from one bee to another bee, or when they are trying to transfer out of the, from one nurse bee to another nurse bee, or from a nurse bee onto a forager so that they can leave the colony. That's the time when they do this. And when they want to be protected and when they want to feed, they wedge themselves into that pocket. So this picture that you see here is not a very good representative of what you should expect. And I think a good illustration of that would be take a look at this bee's hairstyle. Do you see this? He has a mohawk. She has a mohawk. It's very interesting. It's like this because the bee is actually slicked down with wax. I know the researcher who took this image. This bee is not alive. That's why the picture looks so good. You got this bee just standing perfectly still with a mite on its back. This bee is dead and it's been held here, and that's another reason why the mite is in such an unnatural position, because it's going to the highest point on the bee's body because it knows this cannot still be a good host for it to feed on. So we really need to use better representative images when we do our work, or we can convince people of the wrong thing.
To tell the audience that this scanning, the name of the scanning app is Bee Scanning, and you can go into an app store and you can actually, of course, pay money for having it. So be careful out there when they offer that for you. Another thing is about now, it's getting very exciting. There was a guy, one of the guys here writing, because we have made lots of leaflets, books, small books for the beekeepers. And I have to rewrite the thing about the Hemolymph thing, I have to say. So you also gave me more work. But tell us something about, you must make those observations. I'm very often asked by the beekeepers, how long time does the mite stay on the Faradic? And now you don't call it Faradic anymore, but let's keep to the Faradic words.
Nice.
How long time do they stay on the bees out in the colony before they go into the cells for?
So we have found that the mites stay on adult bees, and this is from several different experiments from different researchers, between 3 and 14 days. And so it can be anywhere as few as just 3 days they will be on the adult bees, but they're feeding during this time, causing a lot of problems and damage, or as long as a couple of weeks. And my guess would be that when they are staying on a host for a very long time, it's probably not a very nutritious host. And so in order to get all of the nutrients that they need to go back into their reproductive phase, they probably have to stay on that host for longer. Foragers in colonies have very little fat body tissue, and newly emerged bees have spent pretty much all of the nutritional energy in their fat body tissue, transitioning from larvae into pupae into adults. And so they have very, very, very little fat body tissue. If you want to feed on bees in the colony with a lot of nutrition, you have to feed on the nurse bees. But in colonies that don't have a lot of pollen stores, that don't have a lot of protein, even the nurse bees won't have enough fat body to allow for the mites to get enough nutrition quickly. And so when they have to stay on the bees for a very long period of time, I think it's because the host is less nutritious. But that needs to be borne out in experiments, and I would like to do those in the future.
This is great. I think it's very exciting. Some of the very short questions from Henrik. Fat body, summer bees, winter bees?
Oh, yes. Fat body is different between summer bees and winter bees. In winter bees, fat body, the tissue is very, very thick. There's a huge layer of fat body tissue in the winter nurse bees, and they need all of this because throughout the entire winter, they are not able to consume protein that they will then use to rebuild the fat body stores that they have. And so these winter bees have to go into the winter with all of the fat body that they need to not just make it through the winter, but to then get all the way through the spring buildup, that early part in the spring before the queen is able to produce the next line of worker bees. So during the summer, even though the nurse bees have a fairly thick layer of fat body, it can be much thicker than that. It is much thicker than that during the winter.
And then this brings me to the next question from Care. He's asking about drones versus worker bees? I mean, we know that they prefer the drone brood
They
for
do
multiplying.
prefer
How is it? Do we see them? Do we see mites on drones in the Faradic
so I have
phase?
seen them. Yes, the brick. Um, I have seen these mites in this phase on the on the drones. I have never. I shouldn't say never. Very, 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 very infrequently have I seen the mites actually feeding on the drones, so actually wedged between the plates. I've seen them on the thorax on several occasions, and oftentimes it seems that they're trying to get to a new host, and so they're riding on one bee to the next one. But they do not seem to prefer to feed on drones, and I think the reason for that is because drones, as adults, have very little fat body tissue.
Okay.
They don't do a lot of the functions within the colony that fat body is needed for, and so they don't need a lot of it. They do very strongly prefer drone brood, and of course that has a lot to do with the amount of time that that cell is capped, and also the drones are a larger host resource, so they can sustain substantially more mites than a worker bee can.
So this brings me actually to the next question: is there? I mean, the mites they're biting hole on the pupae behind the seal brood. Do you think there could be any connection on that they prefer those bees? They emerge and they come out and there is already a hole in there. Would it make sense that the mites kind of can recognize the weakened bees where they can go, and or is it all new holes? I think this is a very theoretical question, but it's from one of the audience.
You know, I had not considered that question.
Okay.
As when they are feeding under the cell, they always feed from the same hole. The female will make a hole, and all of her offspring will feed through that hole as well, and it does not close up. But when they emerge as adults, because they don't have that constant feeding of the mites on them, as far as I know, as best I understand it, that hole is closed then. I'm not sure if they're able to identify bees that have been fed on by either themselves or another mite, and then whether they would then gravitate to those bees. But because I don't know if that's the case. I've never seen any studies that have attempted to verify whether that is or is not the case. So if you guys have students sitting around looking for a nice master's or PhD project, you might want to point them in that direction.
In Maryland.
Yes, in Maryland.
Okay.
We're there.
But this is really also bringing me to the next question. I mean, if I could imagine if there would be a parasite on my body and it's Sir Henrik again, and it's sucking my blood, I would survive. But if there would be a parasite biting hole and eating from my body, I would get so weakened. So what does this mean? I mean,
so the organ that the mites are feeding on inside the bees, it has a lot of similarities to the human liver. So imagine if a tick the size of your hand crawled onto your body and slid down to your abdomen and sucked out your liver and then walked away. That would cause a lot of problems inside of your body because there are important functions that your liver does that would not then be done. Inside of the bees, every single one of the functions of the fat body, all 9 of those things that I showed you earlier, every single one of them is impacted negatively by the mites feeding. Where's that chart? And so it's very difficult for the mites to continue being healthy with growth and development, nutrient storage, pesticide detoxification, water loss, immune function, temperature regulation, metabolism, protein and fat synthesis, and vitellogenesis, all negatively affected by this mite. And while their feeding doesn't cause all of these processes to stop, because there is still some fat body tissue that is left, because it has broken down a lot of fat body tissue, it interferes with some of these. It inhibits some of what they do. So it makes these processes less efficient, and some of them just don't get done properly. Oh, I think you need to turn your microphone back on.
I was just reading, so I'm sorry. There's another thing. And again, it's Henrik asking. He's good at making good questions. Do we have, with your findings, does this have any kind of influence on how fast the mites actually are multiplying, since they can eat outside and they eat inside again? Are they yeah, does it have any influence on how fast they multiply? Yeah.
Uh, I did not understand one of the words that you said. You said how fast they what?
The Varroa mite will multiply. This is always a
Ah,
question. It's always this question, you know. I really, when I work with the beekeepers, we really have had some years where we could see the mites are multiplying much faster than they are in other years.
yes.
Yeah.
I'm currently working on a study right now to determine because what my work so it's not shown in this presentation because the presentation would be way too long for me to include everything from my thesis in one presentation, unless you do it in 3 minutes, and then I'll just say, well,
Yeah.
the um, the the reproductive processes of the mites are very heavily impacted by the nutritional content of the fat body. They're actually putting um, nutrients directly from the fat body into their own eggs, and it allows them to produce these eggs very, very, very quickly. And so the mites only have 30 hours to produce the next egg, but the egg is huge. It's between 30 and 40% their body volume. And so to create an offspring that large every 30 hours is a really substantial feat. If you have a resource that is very, very, very nutritionally dense, oftentimes the mites can produce very quickly. And so I would hypothesize that in a colony with a lot of protein, with bees that have a lot of very healthy, very large fat body tissue, even though the bees seem very healthy, if there are mites getting into that colony, they can actually reproduce more quickly in that colony than they would be able to in a colony where the bees are less healthy. They do not have as much protein and as much fat body tissue. I believe as a result of that, the growth of the mites would be inhibited based on the work that was shown in the last chapter of my thesis. Now, I have not shown that direct correlation as of yet, but I've shown some work that is very suggestive based on what they're taking out of the fat body and how much of it they need to put into their eggs in order for the eggs to develop quickly.
So questions like just coming, evolving. This is Christian. He's actually making an interesting question. We know the Varroa mite evolved in Asia, Indonesia. We have Apis cerana and tolerant on la la la. The mites there, they must be doing the same. I mean, eating holes on the
They
adult
are.
bees as well.
So that
Give
is
some
why I
information.
went to Southeast Asia for the last to work on some that question for one of the chapters of my thesis. I only had 3 months to do this, so there was not as much time to try to answer every question, but I wanted to look at how the bees tolerated the mites, specifically Apis cerana, because Apis cerana is the original host for Varroa, but also Apis dorsata, because both of them are much better at dealing with Varroa than Apis mellifera. And there were a few interesting things that were immediately very apparent. One, Apis cerana is really, really, really good at grooming. It much better than Apis mellifera, but in ways that are really interesting because they actually do a grooming dance. We know of the dances that our European bees do with the round dance and the waggle dance. There is a grooming dance that the bees perform. When they get into the colony, they do this weird shake back and forth, and they'll do it around other bees to get the others to pay attention to them. And those bees will then dogpile onto that bee. They do this dance when they feel that they have a parasite on them and they haven't been able to remove it themselves. And the other bees will dogpile on them and they'll remove that parasite and they'll bite it, they'll kill it. And so it's it's something that you never see in Apis mellifera, but it's very helpful for making sure that the phase that is on the adult bees is immediately removed. Of course, Apis cerana has very high hygiene genes. They are able to smell the mites reproducing below the capping. They're able to remove the capping and remove the mites that is built into their DNA. They're very, very, very good at that as well. And they do this primarily, or actually pretty much only, in worker brood. They do not spend their time policing the drone brood. And of course, drone brood is very seasonal. And so the mites are not able to reproduce year-round in the Apis cerana colonies. And so they're just not able to build up to really high levels. They're able to reproduce pretty much year-round in Apis mellifera colonies during the entire growing season, because when they're not in drone brood, when there isn't drone brood present, they can go into the worker brood. Apis cerana does not tolerate that. They've brokered some sort of deal where they've let the mites know, if you hang out in the drone brood, we won't murder you. But if you get into the worker brood, we will kill you and all of your offspring, so don't do it. And the mites seem to be pretty smart at sticking to that deal.
I think we're getting to... Now this is the... We asked for the link for you 3 minutes, this competition for the 3 minutes talk. So there's the link, so everybody will watch that afterwards. I mean, at the end here of this very great talk, because I could continue all night, is really, could you give some thoughts? I mean, Varroa mites, virus, making holes into the bees, trigger the virus, and that was always, for me, Hemolymph. It means it spreads very fast within the bee. Now suddenly it's the fat body instead of, and for me, fat body is something that's not spreading fast. So how?
Allow me to explain.
Yeah.
So remember that the hemolymph is still distributed throughout the bee's body. And so even though they're feeding on the fat body tissue, and the fat body tissue is what they need, when they release those salivary contents into the bee, if they have viruses into their body, that's released into the bee as well. That's going to be in the blood. It's also going to be in the fat body. We have found, sorry, I'm about to sneeze. Excuse me. We have found viruses both in fat body cells, but also distributed throughout the hemolymph. And one of the big problems with, I would say the biggest problem with how this impacts the viruses is that the fat body tissue produces the first line of defense when the bees are impacted by a microbe. The fat body releases these small proteins called antimicrobial peptides. The antimicrobial peptides interfere with bacteria, fungi, viruses, with them moving and replicating. They can actually damage the bacteria, and the fungi and viruses. But the most important part of them is that they flag these microbe invaders as something that needs to be destroyed, and then the immune cells from inside the blood will go and they will attack that virus or that bacteria. If you do not have antimicrobial peptides, the rest of the immune system does not function properly. You kind of break down the rest of the system. And so by these mites feeding on the fat body tissue, they have then created a system where that bee is immunocompromised. And so even viruses that were already present inside of the bee's body, but were held at bay by the bee's own immune system, those can then proliferate out of control. It's been shown that the bees had deformed wing virus before Varroa Destructor came along, before Varroa Destructor caused problems. But when Varroa Destructor feeds on the bees, for some reason, and I think we understand what that reason is now, the viruses proliferate out of control. They have much higher titers if they've been fed on by Varroa, and it causes substantially more damage and substantially more symptoms than it would otherwise. So this is very, very, very closely connected to immune function. Oh, can't hear you again.
Sorry. Okay, thank you very much. This was a great talk. It's really strange with an online seminar. We cannot make any kind of, that's how it is. People are there. But I was so excited about this talk, and yeah, we went over the time, and who cares? It was just great. I'm sure people are making comments now that they're very happy about the talk. So I just want to, they say super, and they make smileys, and they make all the other things. So thanks a lot taking the time.